chapter, the cell, the unit of life, that is endomembrane system. Now, endomembrane system consists of membrane bound organelle whose functions are coordinated. And if we talk about this type of membrane bound organelle whose functions are coordinated, this include number 1, endoplasmic reticulum, number 2, Golgi complex or Golgi apparatus, number 3, lysosome, and number 4, that is vacuole. Okay, so these 4 membrane bound organelle have coordinated function. So first we talk about ER in detail, then we talk about Golgi complex, lysosome, and vacuole. So now first we start with endoplasmic reticulum. This endoplasmic reticulum was first noticed by Porter and Thomson independently. So independently both scientists had noticed this. Name ER, it is named ER by Porter. Now it is present in eukaryotic cell and in cytoplasm it is a complex network of interconnecting membrane bound structure. Membrane bound structures. Okay, so this is single membrane bound structure. It is a single membrane bound structure, and this endoplasmic reticulum can extend from nuclear envelope to plasma membrane. So we can note down all this point and we can understand this all point through the diagrammatic view. So next we talk about a diagrammatic view of endoplasmic reticulum. So now we talk about structure of endoplasmic reticulum. Structure of endoplasmic reticulum. Now if we draw the structure, then this is the nuclear envelope, and from the outer layer of nuclear envelope this endoplasmic reticulum arises, and this form cisternae like this. this cisternae get connected to the another cisternae like this. Okay, and this continues. This continues till plasma membrane arrives. So it connect with plasma membrane at one end and originate from the outer. Okay, so this is chromosome and this is nuclear envelope. This is nuclear envelope, and in this is the outer layer of nuclear envelope. From the outer layer of nuclear envelope, this particular endoplasmic reticulum arises. Okay. Now in this, apart from this, they have some tubule-like structure and in cross section you find here some of the tubules like this. This way we find a tubule, and from tubule vesicle originate. Okay, so like this particular structures occur. So these are cisternae, these are tubules, and this here you can see these are the vesicle which is arising from tubules. Now the cisternae are found associated with the ribosome like this. Okay, and these are ribosome which are found associated with this, and they are ATS ribosome. Now this region where ribosome are present, the region of ER where ribosome are associated with the cisternae towards the nucleus, that is known as RER. RER is rough endoplasmic reticulum, and this region where ribosome are absent, that is known as SER, that is smooth endoplasmic reticulum towards the plasma membrane. Okay, so this way particular structures are associated, and this consists of three structures, that is the what cisternae, tubule, and this. Now this space, this is a cisternae actually this is cisternae. Okay, and this space is known as lumen. The space which is present within the cisternae is named as lumen, and they here through this way the particular one cisternae is connected with the another cisternae. So I hope you are getting the particular clear picture of this endoplasmic reticulum in this way. Okay, so we have dealt with all the structural aspect. Now we have to understand one by one this structural aspect. So first we start with the cisternae, then we will talk about tubule and that all things. First we talk about attachment of ribosome. Suppose this is a cisternae, and if ribosome is attached, ribosome attached through the large subunit. This is large subunit, and this is small subunit. Now large subunit, through large subunit this ribosome attached, and this ribosome are 80S ribosome, small subunit is 40S, and this large subunit is 60S. Here they are attached to ER through this glycoprotein which are known as riboforin 1 and 2. These are the glycoprotein through which large subunit of ribosome is attached with the endoplasmic reticulum. Now, next we talk about the ER compartmentalization. ER divides cytoplasm into two parts. One is luminal compartment and another one is extraluminal compartment. Now, luminal compartment is within ER and it is bounded by ER membrane, that is luminal compartment, and extraluminal compartment is a outer region to ER membrane of cytoplasm. So this way, particularly this ER is divided into two parts, that is luminal and extraluminal compartment. Okay, the luminal is inside the ER and extraluminal is nothing but a cytoplasmic part which is outside the ER. Now next we come to the structure. Structure consists of three parts. Actually it is known as ultrastructure, that is a detailed structure. Now this ultrastructure composed of three kind of structure. As we have seen in case of diagram. Number 1, cisternae. Number 2, tubules. Number 3, vesicles. If we talk about cisternae first, the cisternae are a long, flattened, parallelly arranged sac-like interconnected structure. Sac-like interconnected structure. These are associated with ribosome because of these are found in cells which are actively involved in protein synthesis. Those cells which are actively involved in proteins, this in which you will find this cisternae. This cisternae usually occur in those cells which have synthetic role. Example, cells of pancreas and brain. Second structural component is tubules. These tubules are branched or unbranched. You can see like this structure. That is a branched tubule and this is the unbranched tubule. So the branched and unbranched structures. and it forming a reticular system along with cisternae and form reticular system along with cisternae and vesicles. These cisternae are free of ribosome, so these are common in ER of cells involved in lipid and sterol synthesis. Okay, third component is vesicles. Third component is vesicles, and these vesicles are oval membrane bound structure. Oval membrane bound structure. They are free of ribosome and they are abundant in pancreatic cells, and these are only ER structure, and these are only ER structure found in spermatocytes. So the only ER structure found in spermatocytes. Okay, these are the three components of particularly endoplasmic reticulum which we have studied. Now, next we talk about types of ER. Now, types of ER are two. First is agranular or smooth endoplasmic reticulum, and another one is granular or rough endoplasmic reticulum. Okay, now if you see difference between this, agranular or smooth endoplasmic reticulum has smooth walls due to absence of ribosome. These are free of ribosome, and it has a rough surface due to presence of ribosome. These are involved in SER is involved in lipid and sterol synthesis, whereas granular or rough endoplasmic reticulum involved in protein synthesis. This smooth endoplasmic reticulum is present towards plasma membrane, and this rough endoplasmic reticulum is present towards nuclear envelope. Muscle cells are also rich in SER, so it is known as sarcoplasmic reticulum. So it is known as sarcoplasmic reticulum. And granular or rough endoplasmic reticulum is found in cells which are involved in protein synthesis, and the example are pancreatic and liver cells. In this granular or rough endoplasmic reticulum is abundant. Now we talk about the functions of SER and RER. So next we talk about functions of rough endoplasmic reticulum, that is RER. First, provide surface for protein synthesis. It provides surface for protein synthesis. Number 2, proteins are processed in lumen and packed in membrane bound vesicle for storage or export. Okay, so this vesicles are known as transition vesicle. Then third, formation of nuclear envelope and plasma membrane formation. Fourth, smooth ER formation. Fifth, synthesis of lysosomal enzymes. So these are the functions of RER. It is involved in protein synthesis, ER formation, nuclear envelope plasma membrane formation, and synthesis of lysosomal enzymes. Next we talk about functions of smooth endoplasmic reticulum, that is SER. Number 1, fat and steroidal hormone synthesis. Number 2, glycogen metabolism. Number 3, detoxification of drug or xenobiotics by using cytochrome P450 protein. Fourth, muscle contraction by release and uptake of calcium. And fifth, formation of organelle like spirosomes and glyoxysome. So for synthesis of this also this smooth ER is used. Okay, so till now whatever we have studied, that is about what? Endoplasmic reticulum. Now this endoplasmic reticulum send its transition vesicle with protein to the Golgi complex. So next Golgi complex is going to receive this proteins or synthesized proteins from the ER. So next we talk about Golgi complex. Second component is Golgi complex. This Golgi complex was named after Camillo Golgi, and Camillo Golgi was the scientist who first recognized in nerve cells of barn owl. Barn owl is not able to see. And next, this is present in eukaryotic cell except means absent in mature sieve tubes of plant, they are absent in this mature sieve tubes of plant. Then mature RBC, of mammals. Number 3, some cells, sperm cells of bryophytes and pteridophytes, etc. Okay, so this in all, this is not found. Golgi body is not found in this part, and it is also absent in prokaryotic cell. Okay, in plant it is called as dictyosome, as it is made up of unconnected cisternae. Unconnected cisternae. Next, so these are some of the features with respect to the Golgi body. Next we are talk about ultrastructure. Next we are going to talk about the ultrastructure. And this is the ultrastructure of Golgi body. Now Golgi body, it is formed of parallelly arranged interconnecting system of cluster of disc-like structures. Interconnecting structure of cluster of sac-like structure. So if we draw the diagram of this ultrastructural part of Golgi complex, we will observe like this. You will find a presence of nucleus, and near to nucleus this Golgi body is present. But when Golgi body is present, its convex face is facing the nucleus, and they have cisternae like this. Okay, so this is convex face or forming face, whereas this is the concave face or maturing phase or trans phase. Now be remember, this forming phase is also known as cis phase. Now in this, these are the cisternae and near to cisternae you will find some tubules like this, as we have seen in case of endoplasmic reticulum. You will find this tubules and some of the vesicles coming out of this. Okay, now if you label it, these are the cisternae and these are tubules. These are vesicles, and this vesicles is a mature protein, and these are somewhat Golgian vacuole. So these are the four components through which the whole Golgi complex is made up of. Okay, so please look diagram properly so you can understand how this is present, and we remember the convex face is towards the nucleus and the maturing face or concave face is towards the plasma membrane, and it is composed of four structures, that is a cisternae, tubules, vesicle and Golgian vacuole. So there are four parts or component of Golgi complex, and we will deal with this one by one. Number one, cisternae. Now the cisternae are present in form of flat, curved, smooth, membranous structure with swollen ends. They have a definite polarity, that is convex or cis phase is towards nuclear envelope and concave or trans phase, which is the oldest, and it give rise to secretory vesicle. Secretory vesicles. Okay, so these are the point with respect to cisternae. So the vesicles which are not received, which are arise or produced by the Golgi complex will be termed as secretory vesicles. Now second structure is tubules. Tubules are small, flat, interconnecting structure arising from periphery of cisternae due to fenestration. Now fenestration means separation or breakdown occurs, and due to which from cisternae these tubules will be arised. Third is vesicles. Now vesicles are large, rounded, sac-like structure. They are present at edges of cisternae in cluster. These are pinched off from tubules. pinched off from tubules. Okay, and this vesicle have mature proteins. And the last one that we are talking about, fourth one, that is Golgian vacuole. When we talk about Golgian vacuole, these are a large spherical vacuole produced from maturing phase, filled with some granular or amorphous substance. And important point is that some of them will function as lysosome, and we have seen this Golgian vacuole in case of diagrammatic part. So these are the four parts of the Golgi complex, that is the cisternae, tubules, vesicle and Golgian vacuole. Now we talk about the functions associated with the Golgi complex. So next are functions of Golgi apparatus. Now the function include first main function, and main function is to process, package, transport and release of secretory proteins. Second, glycosidation of lipids and glycosylation of protein. Now glycosidation of lipid means association of carbohydrate with the lipid, so it form glycolipids. But when glycosylation of protein occurs, here carbohydrate associate with protein and it form glycoprotein. So glycolipid are formed by the glycosidation, and glycosylation of protein form a glycoprotease. Third, acrosome or tip of sperm is modified Golgi apparatus. Fourth, root cap cells are rich in Golgi bodies which secrete mucilase for lubrication of root tip, Fifth, it helps in recycling of broken plasma membrane during endocytosis. And sixth, most of protein synthesized by ER are modified by Golgi bodies. They are modified by the Golgi bodies. Okay. So these are the functions of Golgi apparatus, and that is all about Golgi bodies. Now this we have studied one important point, that some of the Golgi and vacuole act as a lysosome. So next coordinated component of endomembrane system is lysosome. Third component of endomembrane system is lysosome. Now lysosomes are also known as suicidal bags. They are also known as suicidal bags. Christian de Duve is credited with discovery of lysosome. These are single membrane bound vesicular structure rich in hydrolytic enzyme which need a water, which are also known as acid hydrolases, and this lysosome contain about 50 hydrolytic enzymes which digest most of the biological substances. Next to that, they have acidic pH, that is 5.65, due to pumping of proton inside. Some hydrolytic enzymes found in lysosome, like acid phosphatases, acid ribonucleases, then deoxyribonucleases, cathepsin, glycosidases. These all are found in particularly in case of the lysosome. It is known that lysosome show polymorphism. Now polymorphism means they are found in various forms. They are found in various forms, so it is known as polymorphism. So on the basis of morphology, their content and function, they are divided into following four forms. They are divided into four forms. Number one, primary lysosome. Now primary lysosome are the lysosome which are sac-like, small vesicle-like structure which are produced from the Golgi apparatus at transphase, and they contain inactive enzyme. Second is secondary lysosome or heterophagosome. So secondary lysosome or heterophagosome are formed when phagosome fuses with primary lysosome. So when primary lysosome fuses with the phagosome, it is known as secondary lysosome, and they contain enzyme against material to be digested. Suppose in primary phagosome there is a protein and we have to digest a protein, then in secondary lysosome the primary lysosome which has a protease enzyme, they combine with phagosome and form a secondary lysosome. So they have enzymes against the material to be digested. That this statement, the meaning of this statement is this. Third type is residual body. Now residual bodies are the secondary lysosome with undigested food material. Okay, so they accumulate this undigested material, and the secondary lysosome will be termed as what? Residual bodies. This residual body pass outwardly, come in contact with plasma membrane and perform ephagy or exocytosis. So through exocytosis, these residual bodies are content is passed out. So they discharge their content outside. But sometimes what happens, this types of discharge does not occur, and residual body load the cells, means they accumulate in the cells and cause aging. Okay, and due to absence of ephagy and loading of this cell, it causes some disorders like polynephritis. They can cause a what? Polynephritis. Number of other diseases linked to malfunctioning of lysosome are other disease due to malfunctioning of lysosome are arthritic joints, gout, and lung fibrosis. Okay, and it also cause some 20 genetic disorders. So it can cause some genetic disorders as well. Okay, so these are the residual bodies. And the fourth component is autophagic vacuole. This autophagic vacuole are formed by union of many primary lysosome around old and dead organelles. So what happens, suppose this is a mitochondria which is damaged or old and it is not functioning, so it may be the harmful for the cell. So what will happen, this primary lysosome will accumulate around this primary lysosome will accumulate around this, and they will join with each other, and after joining with each other they form a vesicular membrane, and when they form a vesicular membrane then they will release all the enzymes inside, and this enzyme will digest this particular dead or component. Okay, so they surround with them with the primary lysosome with vacuolar membrane and digest them. So this is known as autolysis or autodigestion. Okay, so it is a fourth type autophagic vacuole in the case of the lysosomes. Okay, so till now we have discussed primary lysosome, secondary lysosome, tertiary lysosome, this is residual bodies, and fourth also remaining part we have discussed, that is autophagic vacuole. Now we come to the last component of endomembrane system, that is vacuole. So last component endomembrane system is vacuole. Now when we talk about this vacuole, they are membrane bound space. It is a membrane bound space in cytoplasm. It contains water, sap, excretory product, and other material which are not useful for the cell. Okay, so it contains this many structures, components. It is bound by single membrane, called tonoplast. So be remember, this is the important term, that the membrane of vacuole is known as tonoplast, and this tonoplast is selectively permeable membrane. Okay, and this tonoplast facilitate transport of number of ions into the vacuole, that is against concentration gradient. Okay, vacuole occupy 90% of volume of cell. It occupy 90% of volume of cell, and this vacuole are of several types, in which first is a pseudo vacuole, which is found in case of bacteria. Then second is contractile vacuole, which is found in amoeba for the excretion. and there are some food vacuoles. There are some food vacuoles which are formed by the engulfing of a food particles, and that are formed in protist, okay, and it are formed by the engulfing of food material. So this is the vacuole, and that is all about endomembrane system. So we have completed with the endomembrane system. Next we talk about another organelle of the cell, that is mitochondria. Now mitochondria is a plural, singular term is mitochondrion. These are sausage shaped or cylindrical structure. They are a sausage shaped or a cylindrical structure. It is a sausage shaped or cylindrical structure, and if you talk about size, its diameter range from 0.2 to 1 micrometer and average is 0.5 micrometer. Okay, length is 1 to 4.1 micrometer. Okay, this mitochondria are not easily visible under microscope unless stained specifically. If they are not stained properly, then they cannot be visible under the microscope. And the number of mitochondria also vary per cell depending on physiological activity of cell. Depending on a physiological activity of cell. In terms of size and shape also show variation. Okay, this divide by binary fission, and it is a double membrane bound organelle. It is a double membrane bound organelle. Now we are going to talk about this double membrane bound organelle, and we are going to talk about now a structural aspect. So we talk about a structure of mitochondria. Now structure of mitochondria is like this. We are taking a cross section structure. We are talking about a cross section, and in this cross section we visualize this type of diagram. In this, if you see, this is a outer mitochondrial membrane, and this is inner mitochondrial membrane. The space which is present between outer and inner mitochondrial membrane, it is known as PMS, that is a perimitochondrial space. Okay, now This is the inner mitochondrial membrane, and these are the folding of inner mitochondrial membrane. This folding of inner mitochondrial membrane is known as cristae. It is known as what? Cristae. So these are the folding of inner mitochondrial membrane. This is a matrix, that is mitochondrial matrix, and when we see this matrix, in matrix you will find one double stranded circular DNA, then you find 70S ribosome, which are similar to prokaryote. 70S ribosome, they have some RNA. So these things are found in case of this particular mitochondrial matrix. And in mitochondrial matrix, they carry out Krebs cycle. They perform a Krebs cycle for aerobic respiration, to carry out a aerobic respiration. So you can see here, this is the Krebs cycle, which is also known as TCA or tricarboxylic acid cycle. Apart from that, you find a electron transport system here. Here you will find a ETS, that is electron transport system. And here you will find on cristae several F0, F1 units. So if we write about this, then we come to know that ETS, that is electron transport system, is located on the inner mitochondrial membrane. Okay, and one more thing, that oxidative phosphorylation carried out by F0 F1 unit. Now we remember this F0 F1 unit is involved in synthesis of ATP, and ATP is formed by the phosphorylation of ADP and inorganic phosphate, phosphorylate and form a ATP, for which the energy which is required, that is gained from the ETS during the oxidation. So we call it as a oxidative phosphorylation. So now we talk about the structure of F0 F1 unit. So in membrane you will find this structure, and this is the nothing but a membrane, and you will find this type of structure. This is a F0 and this is a F1. Okay, so here H plus pass through this and some energy get released, that is used in phosphorylation of ADP, that is adenosine and phosphate, and ATP formation occurs. So ATP formed by the phosphorylation of ADP, and this is the F0 F1 unit. Now this F0 F1 particle is also known as Fernandez-Moron particle or oxysome. We also called oxysome or elementary particle, which are involved in synthesis of ATP, and due to synthesis of ATP by mitochondria is known as powerhouse of cell. So that is the structural aspect and functions of the mitochondria. Now after mitochondria, next we talk about the plastids. So next we talk about plastids. Plastids are also membrane bound organelle which are found in plant cell and in some protist like eukaryotes. Okay, they are easily visible under microscope as they are large sized. Okay, they have specific pigment, and based on the type of pigment, these plastids are of three types based on type of pigment. Number 1, leucoplast. Number 2, chromoplast, and number 3, chloroplast. Now leucoplasts are colorless plastids, and these leucoplasts are the colorless plastid, so they do not bear pigment and they carry out storage of food. So function is storage of food. Now these leucoplasts are of three types. This chromoplast and leucoplast we will deal after this. These leucoplasts are of three types, that is amyloplast, aleuroplast, and elioplast. They are known as amyloplast because they store carbohydrate, example potato and wheat. Aleuroplast store protein, and elioplast store fat and oil. So fat and oil is stored in elioplast, aleuroplast store protein, and amyloplast store carbohydrate. So this is the function of storage is of leucoplast, which is a colorless plastid. Now after completing this leucoplast, we will now discuss about the chromoplast, then we will talk about the chloroplast. So second plastid are chromoplast. Chromoplast are colored plastids, and they are colored plastid due to presence of carotenoid pigment. Now this carotenoid are of two types. Number 1, that is carotene, and number 2, that is xanthophyll. This carotenoid give yellow, orange or red color to plant part. Be remember, these carotenoids are fat soluble pigment. They are fat soluble pigment. Okay, this chromoplast can be derived from leucoplast or chloroplast, because if you take an example, young fruits are green, but ripened fruit are colored. Or if we say a chili that is also green when it is young, but when it get ripened it is become colored. This is due to conversion of chloroplast into chromoplast. So that is all about the chromoplast, colored plastids which have a fat soluble pigments characteristics. Next we talk about third type of plastid, that is chloroplast, and which are the one of the important pigment. Now this chloroplast, if we talk, these chloroplasts are the greenish pigment. They are the greenish pigment. Majority of chloroplasts are found in mesophyll cell of leaves. They are of different shape, like these are can be lens shaped, oval, spherical, discoid, or even ribbon-like organelle. They have a variable length like 5 to 10 millimeter length and 2 to 4 millimeter width. Okay, their number varies from 1 per cell to 1 per cell of Chlamydomonas green alga to 20 to 40 per cell in mesophyll. So they are 20 to 40 per cell in mesophyll cell, but they are 1 per cell in the case of Chlamydomonas. So number also can vary this much. Okay, so these are the some of the points with reference to chloroplast conditions, shape, size and occurrence. That is the all thing. Now we talk about a structure. Structure of chloroplast. These are double membrane bound organelle. So if we draw the structure of this double membrane bound organelle, it looks like somewhat this. This is the outer chloroplast membrane, and this is the inner chloroplast membrane. Now what happens, the folding of inner chloroplast membrane occurs and this form thylakoids. Okay, so here we see this type of structure which are known as thylakoid. Yes or no? These are thylakoids. So in this also, if we see, there is a space within them. And if we label this, then we find that this is the outer chloroplast membrane. This is inner chloroplast membrane. Then these are grana thylakoid. They can be called as a thylakoid only, but they can be specifically called as grana thylakoid because they are present in whole granum. So this completely a stack of thylakoid is called as granum, and plural term which is used for that is a grana. Now this is also a thylakoid which are named as stroma lamella, and they can also be called as stroma thylakoid. This is the stroma, and when we see in case of stroma, this stroma has double membrane bound, double stranded circular DNA. They have some RNA as well, and they have ribosome as well, and that ribosome are 70S ribosome. So we will study ribosome after this chloroplast, but you be familiar with the structure, which is important. It's a double membrane bound structure. The folding of inner membrane occur and thylakoid will be formed. Thylakoid are of two types which are present in the form of grana, that is known as grana thylakoid, and the stroma thylakoid or stroma lamella which connect the two granum, that is known as stroma lamella. This is also named as fret. They are also named as fret or fret channel. So they have a ribosome as well, 70S, and there is what particularly double membrane bound double circular DNA. So this is the thing. Now this chloroplast play a role in photosynthesis, where light reaction So they play a role in photosynthesis, in which large reaction occur on membrane of thylakoids, that is both grana and stroma thylakoid, and dark reaction occur in stroma or matrix of a chloroplast, where direct reaction is performed. And in light reaction, synthesis of ATP occur. That ATP are used in dark reaction, and in dark reaction glucose formation occurs. Okay, so this is the what a structural part and functional part of photosynthesis. Their stroma bear a double stranded circular DNA, and they have 70S ribosome. So this is the structural part of chloroplast. Now we have studied chloroplast and mitochondria both, and we have seen some of the similar structure. So chloroplast and mitochondria are endosymbiont. They are endosymbiont and they show symbiotic mechanism, and they have originated from bacteria, means they show bacterial origin. So why it is considered that they have endosymbiote and they have a bacterial origin? Because both are double membrane bound organelle. Next is they both have 70S ribosome. They have their separate RNAs. Third, both have double stranded circular DNA. Fourth, presence of porins in outer membrane of chloroplast and mitochondria. Now porins are large sized protein which can allow transport of some small protein, so that is the thing. And they have their own ETS. So they have their own 70S ribosome, they have their own double stranded circular DNA, they have presence of porins, and they have particular ETS. These four are the more important point which show that chloroplast and mitochondria have originated from bacteria, and it is might be like that bacteria has gone within the eukaryotic cell and permanently it symbiotically survived within the eukaryotic cell. So because of this it is an endosymbiont. Okay, so this is the with respect to chloroplast and mitochondria. Now we talk about the next topic in this lecture, that is ribosome. Ribosome are membraneless organelle. They are membraneless organelle, and ribosome are also known as Pallade particle. Known as Pallade particle because it was first observed by George Pallade in 1953 under electron microscope. It is a granular structure involved in protein synthesis. It is involved in protein synthesis. Composed of RNA, that is specifically ribosomal RNA, a ribonucleic acid, and protein. Now this ribosome are of two types, 70S and 80S. 70S found in prokaryote chloroplast and mitochondria, whereas ATS found in eukaryotic cytoplasm. So they are made up of rRNA and protein. They are of two types, 70S and 80S. Now first we talk about a 70S ribosome. 70S ribosome has a two subunit, that is 50S and 30S. So if you draw the structure, suppose this is a small subunit and this is a large subunit. Okay, so this is 50S and this is 30S. Now in this it is made up of rRNA and protein. So in this you will find 16S rRNA, and here you will find 23S rRNA and 5S rRNA. So these are the rRNA which is present in large and small subunit. Now here S represent sedimentation coefficient, and it is also known as sedimentation coefficient. Sedimentation coefficient describe the time they take to sediment, and 1S is equal to 1 into 10 raised to minus 13 second, one sedimentation coefficient. And this sedimentation coefficient was discovered by Swedberg, so known as Swedberg unit. It is also known as Swedberg unit. Now we talk about ATS ribosome. When we draw a structure of ATS ribosome, this is 40S and this is 60S. Now in 40S we find a 18S rRNA, that is a type of rRNA, and in this you will find 28S rRNA, 5S rRNA and 5.8S rRNA. Okay, this type of rRNA are found in case of this. Now this ribosome, every ribosome have three sites. E, P and A. They have three sites, E, P and A. E stands for exit site, P stands for peptidyl transferase site, and A stands for acceptor site. A for acceptor of amino acid, P peptidyl transferase, this is transfer of peptide, and E for exit of tRNA. Okay, that is now might be you will not understood, but in 12th you will understood how this particular three site work. Okay, in which P is common to both large and small subunit, and E and A are associated with a large subunit. Okay, now this subunit can be dissociated to remain associated ribosome subunits. There is a need of 0.0001 molar concentration of magnesium in cytoplasm. So this 0.001 molar concentration is there, then only this subunit can associate, otherwise they found to be dissociated in a dissociated form. And these are the ribosome, and we know the function of ribosome, that they are involved in protein synthesis. We have discussed with respect to ribosome one more structure in prokaryotic cell, that is a polysome, and we know that polysome is the mRNA with the multiple ribosome. Okay, so this is all about ribosome, and that is all about this lecture. Now in next lecture we will talk about the remaining organelles of a eukaryotic cell.